8. DNA, INB. What is the nature of science? A scientist's everyday activities involve testing ideas and theories, making calculated changes, and generally trying to make sense of the natural world around them. We scientists live by the following ideas. The natural world is understandable. Science is based on evidence. Science is a mix of logic and technology. The scientific community works together and shares research. Scientists try to stay open-minded. And scientific theory often changes when new information surfaces. Type it. Which shape is which? Helix or double helix? This shape here is a single helix. This shape here is a double helix. You see the two strands. Drag it. DNA or deoxyribonucleic acid resides inside the nucleus of every living cell. It was discovered in 1869, but the structure remained a mystery. In 1952, using X-ray photography, Rosalind Franklin observed DNA, but could not identify the shape. A year later, Francis Crick and James Watson used her images to describe the twisted ladder or double helix structure of DNA. Dr. Rosalind Franklin and her photo 51. Watson and Crick and their double helix model. So here's their model here. They just look sketchy because they stole her image. Type it. DNA contributions timeline. I'm going to read over these first. In 1866, Gregor Mendel, we're going to learn about him later, and the heredity of peas. Interesting. 1944, Oswald Avery, first evidence that DNA carries genetic information. We're not going to learn about him. Actually, you just did, so you're welcome. Erwin Chargaff publishes How Reproduction Involves Chromosomes in 1950. I'm not going to learn about him. 1952, Rosalind Franklin takes photo 51 with X-ray diffraction machine. James Watson and Francis Crick just a year later, sketchy, published The Structure of DNA Double Helix. 1961, Marshall Nierenberg, cracking the genetic code. So he was able to sort of read and saw the different bases and said, oh, they're in a code and there's only 4. Wow. And what they meant. Alec Jeffreys developed DNA fingerprinting. That's cool. That's a year before I was born. And that means like what cops do, and they can use DNA to identify a person. It has, it's essentially better than a fingerprint. Because it's 99 point something % accurate. 2000 to 2003, that's 3 years. Scientists mapped the entire human genome and found all the genes in each strand of DNA in a human being. That they literally mapped out the entire set of human DNA. So all the chromosomes were, mapped out, all the DNA strands. All right, now for the questions. This is a type it, so you're going to type your answers. Who took the first picture of DNA? The first really good picture. That was Rosalind Franklin. Who made the first model of the DNA of the double helix structure? And that was James Watson and Francis Crick. Who figured out how to read DNA? Who was able to crack the code? That was Marshall Nierenberg. And how many years did it take scientists to map every human gene? See if I get you at this. 4 years. Count it. 2000, 2001, 2002, 2003. 4 years. Y'all said 3, didn't you? Label the ladder. What part of the ladder is the rail of the ladder? And what is the rung of the ladder? What is this arrow pointing to? Oh, man, I did it wrong. That is pointing to the rails, and this is pointing to the rungs. Using the picture to match the correct statement to each term. DNA, gene, chromosome, allele. Let's read this one first. It says the coded instructions that store and pass on genetic information from one generation to the next. So this is coded instructions and it passes on genetic information. This one says sections of DNA that carries a code for a particular trait. So it's a section of DNA. That right there is going to tell you what it is, the word section. This one says a strand of tightly wound DNA. We know what that is. When you tightly wind DNA, what does it make? We'll go there in a second. Different versions of the same gene. So we know if there's different versions of the same gene, like blue and brown eyes, you both have the eye gene, the eye color gene, but they have 2 different types of the same gene. We'll talk about that in a second. So, first one, the coded instructions that store and pass on genetic information. That's right, that's DNA. Now let's do this one. Sections of DNA that carries a code. So a section, look down at this picture. Where's there a section of DNA? Oh, look at that. It's a section of DNA. That's a gene. Is this in order? This one says a strand of tightly wound DNA. We know that if we wind up the DNA, like in this picture, really tightly, it will form a chromosome. Nice. And the last one, it's definitely in order. Different versions of the same gene. So we know that this little picture is a gene right here, this little green spot on this chromosome. But not everyone's, let's say, eye color gene is the same. So the different genes wrapped up in the chromosomes, we call those alleles. Next. All right, so we're going to label these 4 things. We're going to label DNA, gene, chromosome, and allele. All right, let's start with DNA. Let's start with DNA. That is going to be down here. This whole strip right here is, just kidding. Let's start with chromosome, shall we? This up here is tightly wound DNA. That is the chromosome. Actually, you go ahead and take a second and label these, and then we'll go over them together. Okay, that's enough. All right, the next one, what is this pointing to? Oh, this little thing right here on the chromosome. What is that? Oh, that's an allele. You guessed right. This is pointing to a section of this DNA strand. Oh, that would be a gene. And like I said at the beginning, this is a DNA strand. Okay. Alrighty. So now we're going to start on this side. And we're going to say what these pictures are first. So let's look at this picture. What is this guy? He is representing, look at those options, chromosome, gene, DNA, ribosome, protein, or nucleus. You guessed it, that is a chromosome. What is this guy right here? This is a protein chain. Good. This guy, we know, is definitely the ribosome. We know this fella. He's the nucleus holding onto that DNA for us. He is the DNA itself. And this is a section of that DNA. That would be the gene. Good. Now we're going to go through this section and figure out which of these descriptions of each it's referring to. We're going to start with this one. It says, I make up all cells. I make up all cells. This is what all cells, tissue, organs, and ribosomes. It says cells, tissues, and organs are made of this, and ribosomes make it. So what do ribosomes make? Proteins. Good job. I translate the code. I translate code from a gene, and I print out the right protein. So who prints out the proteins? Who translates that DNA code? That would be the ribosome. And next, if you wrap an entire strand of DNA up really tightly, you will get me. So if you wrap up an entire strand of DNA really tightly, what do you get? You get a chromosome. Good. Next, I am one section of DNA. I hold the code to make a protein for a trait. So again, one section of DNA is a gene. Good. It says I hold all of the genetic information for an entire organism. All of the information for an entire organism, that's in the DNA strands. Okay? Last one says I keep the DNA safe inside me until it's needed. That's our nucleus. He's so cute. I need a sip of coffee. Hold on. All right. Let's focus on these little fellas and ladies or whatever up here. Look at their eye colors. All right. We're going to look at their eye. Oh, look at the little chromosomes above them. Notice something? Oh, look at that little thing that's wrapped around each chromosome. Oh, they're different colors. Interesting. Different versions of genes alleles look. What? Different versions of genes, alleles. Look at all those beautiful alleles. Oh, I'm sorry. This is drag and drop. And this is the pile of the different things you'll need. So let's do this. The first one goes down here. All right, that's the next one goes here. If you put these in the right order, they'll make us like a paragraph, and I'll read it to you. So just take a second and try it, and you're going to just place these in the right place while I'm doing it. And these should be in the same order as yours. All right, now I'm going to read it to you. I'm a chromosome. That pink thing wrapped around me. Oh, this guy right here is talking, by the way. I'm a chromosome. That pink thing wrapped around me is a gene. It looks like this when the DNA wraps up tight to make me. This pink gene is the eye color gene. Everyone has one of these genes, but not everybody's is the same color. All of these kids in the image above have an eye color gene or their chromosome, but they all have different versions of that gene. We call that different versions of the genes alleles. That makes so much more sense. Look at all those beautiful alleles. Each chromosome has many genes. We just highlighted the ones to explain the alleles. Rope analogy. Okay, so there's 3 pictures here and 3 descriptions. And one of the pictures of ropes is representing DNA. One represents a chromosome and one represents a gene. So let's look at this first picture. This picture is a strand of rope tightly wound up. What does that sound like? Tightly wound up, right? That sounds like a chromosome. Look at this. This is a bunch of just unraveled rope. Just a lot of it. A whole lot of unwound rope. Yep, that's DNA. And this, oh look, it's sections of rope. Sections of rope. There we go. That's a gene. All right, so this one says long strands of genetic material that holds thousands of genes and is in the shape of a double helix. If you actually look at the rope, did you notice that rope is a double helix? It's crazy. And some ropes actually have like 5 different helixes, but we're not going to talk about that. Let's just pretend that this only has 2 strands and it's a double helix. Sections of DNA that hold code for one trait. Again, it says sections of DNA. We should know that by now. That code is sent to the ribosome and makes the protein it codes for. So we know that anytime it's talking about a section of DNA, we're talking about a gene. When DNA is wrapped up tightly, again, every time it talks about wrapping up DNA tightly, we're talking about a chromosome. When DNA is wrapped up tightly in a well-organized thread-like bundle, it keeps the DNA from getting tangled up. The other strands, I definitely did a bad job with this one. It should say in the other strands or with it. Just go with it. Oh, I made a video for this. Watch. So, I'm going to pause it actually. A chromosome is DNA wound up tightly. Imagine 46 long strands of rope being kept in one container. It's crazy. If you don't organize them, somehow they would all become tangled and knot up. Just like that picture of all those ropes. Chromosomes are our cell's way of keeping our DNA neat and organized. So we're going to use the scribble tool to make chromosomes out of tightly wound DNA strands in a box to the right. So we're going to draw a chromosome right here, and you're going to go up to the top screen. It's going to show it here in a second in the video. The scribble tool. You're going to go to the line tool, go down to scribble, and you're going to scribble it and do it in the same way as the video. Watch. Okay, so go to the top, go down to scribble, and just like how chromosomes are made, we're making these like scribbly loopies, okay? And we're going to do it in the shape of a chromosome. Look at that. Nice. You think about it, a chromosome is like we're using a lot of line there, right? If we were to take that line and stretch it out into one long line, that's a lot of lines, right? But we're going to, it's the same way if you see someone with curly hair and they pull one curl really straight, it's way longer than it looks. Alright, so you're making a chromosome out of your little curly, oh, beautiful. That's what you're doing. Gorge. Alright. Now I gotta go to the next slide. Turn. Come on. There we go. Alright, so we've talked about the different parts of DNA. We're going to start, I really don't know where it's going to start, so we're just going to go for it. I think it's going to be this thing. This thing, this 5-sided figure here, is the sugar, yes. And this thing right here, alright, this is part of the backbone, by the way. Oh, I think that's the next slide. This is, oh, what did I do? Oh, we're going down this row. Guanine. If you look up here, it has a little key. Guanine is the red base. And the next one is cytosine. It's the blue one. And these aren't the actual colors of the DNA bases. This is just for this notes project. Hydrogen base or hydrogenous base, that is here. Nope. Oh, what am I doing? That is here. Teachers make mistakes too. Yep, these are the bases, the 4 bases. Thymine, again, yellow. Adenine is the purple. And phosphate, there she is right there. There we go. Phosphate, part of the backbone. Last but not least, trying to make it feel better about itself since I forgot what it's called. All right. Now, this was a daily warm-up one day for us. This right here, this little part of 1 phosphate, 1 sugar, and 1 nitrogenous base is, man, alright, so we know that this is the deoxyribose sugar. The nitrogenous base is here. We know this is the phosphate, and we know that this right here, this all thing together, is a nucleotide. Alright, next. So, hydrogen bases pair up in specific sequences. Adenine always pairs with thymine. Cytosine always pairs with guanine. And this is how I remember it. Apples grow on trees and cars park in garages. All right? Apples on trees, cars in garage. Everybody say it. Apples on trees, car and garage. Good job. Now, the first one is purple. All right? Purple, that's adenine. What does adenine pair with? Apples grow on trees. That's thymine. We're going to place a thymine. What am I doing? All right. Same thing. Thymine. This is thymine. What does it pair with? Pairs with purple. All right. So next one is blue. Blue is cytosine. What does cytosine pair with? Car and garage. G. G is red. So blue goes with red. Next. Yellow. Yellow is thymine. Apples grow on trees. So yellow goes with purple. So you don't have to remember these colors. You need to remember the letters. That A goes with T, C goes with G. All right, next. Guanine, right here. Guanine is garage. And what goes with garage? Car goes with garage. So we're looking for the blue. There she is. Beautiful. They're pairs. Hydrogen bases pair up in specific sequences. And so this is really important for DNA. They only pair up with their partners in DNA sequence. And so that's part of the code that one theorist, scientist cracked. All right. Next. Use the base letters below to complete the base pairs on the DNA model to the left. So what we just talked about is car, garage, apple, tree. All right, so the first base is A. That's adenine. What does A go with? Apple. You're looking for tree. So you're going to get this and drag it here. Take a second and see if you can get the next couple. We'll do a few of these together. Car goes with garage. Good. Garage goes with car. Garage again goes with car. Apple, tree. Apple, tree. Oh, man, I'm repetitive. Apple, tree, tree, apple, tree, apple, car, garage, garage, car, tree. That's right, apple. Sip of coffee. All right. So, look at this and look at this. This is only half of a strand of DNA. This is the original strand, and this one has been mutated. Probably by nuclear radiation. No, the original strand of DNA is seen on the left, right here. On the right, you see the same strand of DNA. However, an error or mutation has occurred. This error could cause a positive, negative, or no change on the protein made from this genetic code. So this is one of those situations. Genetic code will have errors. They will have mutations. They often do. Sometimes it has a positive effect. It could make someone's heart strong. It could make someone's hair longer, or it could give someone a birthmark, change their voice, make them tall. It could have a negative effect. It could give them a disease. It could cause them to, have a deformity or discoloration of pigment that could be sensitive to sun, or it could have no change at all. So you can have two different strands of genetic code. One has a mutation, but it still functions the same way. All right, so this is one long strand of DNA. One very long strand of DNA. How many genes, and remember genes are sections of DNA, how many genes are in this strand of DNA? And just so you know, in a real strand of DNA, there could be thousands of genes in one single strand of DNA. So this isn't accurate, but this is just us practicing what is a gene and how are they kind of divided. And so, all right, let's count these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So you're going to type in this box right here, 18. Now remember, once it's wrapped up around a chromosome, and again, there's actually thousands of genes here, but for the sake of me showing you this, this is a model. It's not completely accurate, but it's great for learning. So once you wrap those genes up in this tightly wound chromosome, the genes are no longer linear, like in a straight line. They're wrapped up in an area. So these genes are represented by like little bands around the chromosomes. How many genes are in this chromosome? So we're going to count them. 1, 2, 3, 4, 5, 6, 7. There should be 7. Oh, I'm so good. Oh, I didn't type it. This is one of my last minute ones, but type it. All right, so we have hair color gene, eye color gene, freckles gene. Oh, there's my mouse. Earlobe gene and pinky finger gene. Which gene is showing more than one allele? So is this one? Nope, that's the same allele. That one, oh, look at that. I even pointed to it. This is two different alleles for the eye color gene. So we're going to type the eye color gene is showing more than one allele. Everything else is the same. Type it. All right, now I'm going to put it all together. Everything we've learned so far. DNA is so incredibly amazing. A human sex cell has 46 strands of DNA in its nucleus. So one sex cell, one cell like a woman's egg or a man's sperm, one of those cells has 46 strands of DNA in its nucleus. One strand of DNA could have up to 1,500 genes coded within it. Each gene sends a copy of itself to the ribosome that will use the code to make that protein. That protein is used to make many things like organelles, cells, skin, and organs. And without these things, organisms wouldn't be able to perform life processes such as growth and repair, reproduction, gas exchange, metabolism, and response to stimuli. Alright, this is the last slide. I'm not going to do the entire thing with you. I'm going to show you how to do it, and then I'll give you the answer for the first one. And I might not make you do this. This is more for fun. But the hydrogen bases form a code, and this is just like this is like the bases from one gene. Okay. Now, are these actual genes? No, I made this set of code up. So, so this is like a gene, a code for a gene. And just like a gene, it's going to make something. Usually genes make proteins. This is going to make a sentence of some sort. It says the proteins made from each genetic code is sent to its location to do its function. Okay, so the proteins that's made are then sent off. So if it's a protein to make heart tissue, it's sent off to form or take its place as heart tissue. Okay, now so this is just code. ATA, what is that? So you gotta find in this thing. Oh, that one's an L. And CCC. Oh, that's E. And the next one's AAT. Oh, G. So leg. And then AGA. That's a space. Leg. These are genes for specific places in the body. So ATA, I forgot what I made. ATA, that's L. And then CCC, that's E. Leg, it's leg link. Alright, so I would recommend, you don't have, I would recommend just typing each letter in as you go. Take your time with this. You can do like 3 or 4 of them. Maybe I'll split the class up and let every group or whatever find one. So that's it. Let's get to work.